Hello, everyone. Thank you for joining the webinar today on 8 ways that using osmolality testing can improve cell and gene therapy process development and manufacturing. My name is Shweta, and I'm the Senior Product Manager at Advanced Instruments focused on driving the biotech business, and I'll be the moderator for this webinar. Joining me today is our speaker, Christina Wright. Christina is an application specialist, an application scientist at Advanced Instruments with a background in drug manufacturing support and molecular biology. She's a motivated researcher and a spokesperson for scientific advancements, as shown by multiple presentations and collaborations with other subject matter experts. Hello, Christina.
Hello, Shweta. Thank you so much for the wonderful introduction.
Thank you. It's a pleasure to have you today with us. So, Christina, what are the goals for the presentation today?
So today what I'd really like to do is kind of start to focus our eyes more on the cell and gene therapy space. So at Advanced Instruments, and especially within this team, my primary goal is to better understand some of the pain points and really understand the processes that go into building workflows within cell and gene therapy spaces, so that we can figure out ways in which osmolality testing can help fill those gaps, solve some of those pain points, and really improve the process overall. So what I'd like to do today is share some new learnings. As you said, we really value our relationships with different key opinion leaders in the field and some of our customers. And we like to share some thoughts on different emerging and more novel use cases for osmolality testing within cell and gene therapy. And really just start a conversation, just start a discussion about what are some thoughts from some of you that are listening. And as we start to investigate a little bit closer and start working more towards getting some projects started and getting scientific data, which ones are really appealing to you and what's most valuable to the customers?
Perfect. So indeed, as you mentioned, we really want to hear what's on your mind, what a part of your process is, and really hoping to have this be a conversation starter to really catapult the value of osmolality and the usage of osmolality in cell gene therapy. So having said that, please feel free to ask any questions during the session, and we'll have a live Q&A session right after the presentation. So with that, Christina, over to you.
Great. Thank you so much, Shweta. So thank you guys again. I'd just like to echo Shweta's sentiments for joining us today. I'd like to start with an overview and kind of a brief agenda of what we'll be discussing today. I'd really like to start with just the foundation, some of the groundwork. What is osmolality? There are a lot of properties that are mentioned and discussed throughout the literature when it comes to kind of describing colligative properties of solutions. So let's just start there with some basics, and then we'll really start to dig into the meat of this, which is how does osmolality testing serve as a key parameter within the cell and gene therapy field? I'd really like to kind of break it down into some upstream applications, downstream applications, and then we'll move more into formulation and final product use cases. And lastly, how can Advanced Instruments help you as we start to really understand what some of these uses are? How do we, or what devices and technologies do we have that can get you up and going? Hopefully, after all of this, you guys will be excited and ready to start on some of these projects and start with some discussions. So with that being said, I'd like to just, as I mentioned, quickly define osmolality. So I mentioned it's a colligative property of a liquid solution. Anytime that solutes are introduced into a solvent, the resulting solution is going to be different in many ways from the initial solvent itself. Osmolality describes just one of those ways, one of those properties. But when we look at it as a definition, it really describes the total number of solutes in a liquid solution. It's expressed as osmoles or milliosmoles, more specifically, of a solute per kilogram of solvent. And the reason it's important to note this is because we're measuring it per mass, osmolality is actually independent of the volume. So it's not affected necessarily by the solution's temperature change, which really makes it, it kind of drives a lot of the reasoning behind why this provides kind of an orthogonal value to some of these applications that we'll be discussing. So I wanted to start off on that note so that we can better understand how osmolality fills gaps and really provide a unique role throughout these processes. So with that being said, I do want to take a quick poll of the audience, if you will. And we're going to start with the first question, which is really, where do you currently use osmolality testing, if at all, within your current workflow? So I'll give you guys a little bit of time to answer that question, and then we will proceed. Great. Awesome. Okay, so we're seeing some upstream, we're seeing final product, which is definitely expected. And then we're seeing that some people are not currently using osmolality testing in their process. So I think that in many ways that lines up with what we would expect to see and what we've seen from our current customers. And it really drives the point of this presentation, which is to kind of touch on some new ideas and get people excited about potentially using your current instruments or new instruments at other areas within your workflow. So thank you so much for participating in that. So where does osmolality testing fit? It sounds like for the most part, we're kind of seeing it upstream. We see it obviously as a final product test, as it is compendial, to identify and make sure that the final composition of therapies is correct. But I wanted to give an overview slide so you can see how many different areas this test really is applicable. And it really does serve very, very unique value with each of these. So as I said, I won't be able to get into each one in detail, but I wanted to introduce some information that's been published and also some information that we've gathered from KOLs and from customers on where they find value within some of these areas. And the idea is that this is just kind of our starting ground. We really want to get the discussion started. And then from here, we will definitely be holding more webinars and providing more data as that comes to light. So let's start with upstream processing. So first of all, I wanted to take a quick look at something that maybe some of you are already using for osmolality testing, or an area in which you're already operating some of our equipment, and that is media quality control. So for media QC, we do know there's a clear trend. This is, within the mAb space as well as within gene and cell therapy. We're seeing an uptick in the number of customers, of companies that are producing drugs that really are purchasing off-the-shelf media. They're outsourcing it and getting it from external vendors. So as a result of that, we really see that quality control around the media, whether it's outsourced or made in-house, is becoming perhaps even more important than ever. Osmolality testing is one area of that quality control that really helps to ensure the optimal cell growth or eventual expansion of your cells that are going to be benefiting from the media that you're purchasing or making in-house. So to get a little bit more specific, it's really important to check the osmolality and also produce some specifications around each media batch to make sure that the environment is of the appropriate physiological range as far as osmolality goes. And I did want to provide a little bit of insight into some publications that really get pretty specific to HEC-293 cells. But what's interesting is that we tend to think of osmolality in general, if it's around the physiological range of, about 290, maybe 300 milliosmoles, how far out of that range can we get? And there's some literature supporting the fact that obviously you don't want to go too high. That's going to potentially compromise your cells. But at higher osmolalities, there is the potential to, if it's desirable, maybe lower the growth of your cells, but increase productivity. If that's something that you guys are familiar with or have experience with, we would love to hear more about that. Feel free to drop us a comment in the question section, and we would love to hear more. But this is definitely one of the use cases that we see is gaining popularity for osmolality testing. So that's media. Obviously, it goes beyond just bulking your media and getting all of that together. So when the cells are in place, whether it's going to be something that's in suspension or you're looking at some adherent stem cells, monitoring the osmolality also becomes critical. So specifically, when we look at stem cell culture expansion, I would say that osmolality really is a critical process parameter here. And honestly, that's across bioprocessing modalities, not even just within stem cells. But because the cells do require a physiologically relevant environment, there's this dynamic interaction as they're absorbing nutrients, they're producing waste. It's something that's going to be changing. The osmolality will be changing over time. So it's something that should be monitored for sure. And if we focus specifically on the expansion of different types of stem cells, we see the same value across cell sources even. So not everything that we find obviously is testing the same type or source of cell in the body, but we have seen that some of these general trends that I mentioned here on the slide. We typically think of osmolality specifications as ensuring that cell viability and productivity are preserved. There have been a lot of studies so far that really highlight the benefits of increasing osmolality, and that kind of plays into the same thing that I said with the media QC. Up to a certain extent, again, it may be possible to slow the growth and maximize the productivity here as your cells are in culture. And developing a specification around this is really key. So this is one that I would say this application is really exciting for us, but definitely very new and emerging. So as we try to gather more information, if it's something you're interested in or have experience with, looking at osmolality within transfection efficiency improvement, please let us know. Again, feel free to drop a comment below, and we would love to have a conversation with you. What do we know so far about osmolality and transfection efficiency? So if we look at osmolality in theory and kind of look at osmosis in cells, characterizing osmolality and osmotic pressure within and outside of a packaging cell could be really crucial to the efficiency of the transfection, as well as ultimately the viral yield from this process. So the rate of in situ complexing, as well as any of the dynamics between the nucleic acids, the DNA, and here I have PEI, but if you're doing a chemical transfection, really whatever that reagent, whatever that solution is, the dynamics of that complex are dependent on osmolality and a number of other key parameters. And here you can see that I did put a source that did specifically looked at PEI to DNA complexes. Additionally, osmotic pressure is believed to be a key factor in the stability of the eventual internalized DNA to PEI complex. So the question really is here, I mean, we see how valuable this can be, and I think in general, there's an understanding that because we're dealing with cells, osmolality is going to play a role here. But how does osmolality testing, how will one of our osmometers fit into transfection protocols? And that's something that we are really working to prove and get some data around. But ultimately, we want to look into the relationship between osmolality and the stability of the complex of the transfection agent, whether it's PEI or whatever, the complex with the DNA. And the goal of this would really be to answer some critical questions specific to that protocol. So when we look at the solutions that are involved, that's where we would measure the osmolality. So what are the media needs? What is the osmolality of the transfection cocktail? And, you know, of course, there's going to be a number of questions around this that we would have to, and other parameters we would have to monitor in parallel. But ultimately, the goal is to provide information on the transfection efficiency and use osmolality to develop specifications and offer a state of process control. Okay, and to, I guess, round out some of our upstream applications and some really exciting areas that we've been investigating recently, I want to talk a little bit about the stability of vectors. And in particular, I'm going to be discussing retroviral vectors. However, there is some literature that discusses AAVs as well. But here I wanted to kind of start, if you see up in the upper left-hand corner, there's a group that's really been looking at the relationship between osmolality and the stability and productivity that they're seeing within their vectors. And that relationship, you know, obviously, as usually happens within academic laboratories, you have a finding and then you kind of dig a little bit deeper into the why. So a relationship was established between higher media osmolalities. You'll notice that trend. We keep seeing that if you go above that typical 290 milliosmoles, you may start to see some positive effects when it comes to yield and productivity. Same story here. So they linked vector stability and productivity at higher levels to higher osmolalities. The question is, why? What's going on internally that would allow this to, or what's going on within the vector membrane to create this trend? So the first piece I'd like to draw your attention to is on the right-hand side of the slide. That is, they found that higher media osmolality, higher osmotic pressure in the media actually induced a lower ratio of cholesterol to phospholipids within the lipid membrane. And that was associated with higher stability in the viral vectors that were eventually produced. So that's one side of it, is looking at that lipid ratio and the makeup and final composition of the membrane. The other piece that was investigated, and another key factor, is sugar metabolism. So increased levels of sorbitol or any type of osmolite, in this case it's a sugar, but it positively correlates to the retroviral stability and the productivity. And I did mention here that in this case, for this lab, for this team, they were using fly cell lines. So again, this is pretty specific to what this team was looking at, but it's interesting that in general, they were able to establish this correlation, positive correlation between osmolality and the viral vector stability. Okay, so as I said, we'll wrap up some of our upstream use cases. If you have any questions, again, please feel free to post them below in the Q&A section in the questions area. And at the end of the presentation, we will make sure that we address all of those. Next, I would like to move into downstream processing. Okay, so similar in a way to the media QC story that I explained earlier, osmolality testing has proven pretty valuable when it comes to buffer QC. And I would say in downstream in general. You'll notice on the poll that we don't have a lot of implementation of osmolality testing within downstream applications. That's across modalities. We see the same thing with protein therapies, monoclonal antibodies. But one thing that we have found, and we do have some data from 2019 that shows that osmolality has proven to be a valuable orthogonal measurement when it comes to representing and measuring buffer concentration for buffers that are really, or buffer components that are really commonly used in downstream processing. So just to take a quick peek at that data, these are just some representative graphs, and it shows osmolality and conductivity across concentration ranges for these buffers and buffer components. One question that I've gotten is, we don't necessarily operate at these high concentrations where you start to see some of the changes in conductivity. And the answer to that is, as we start to see a drive towards more continuous processing and downstream processing, and we start to see these more concentrated buffers being produced and higher increased implementation of things like inline buffer dilution systems, we do really come across some solutions of buffers that are really highly concentrated. So we wanted to make sure that we covered the full range. And as you can see from the graphs, while not perfectly linear, osmolality certainly offers really great predictability and reliability across the concentration ranges. Another thing that's really interesting is that the resolution of these curves is great. So if we're operating from 0 to 2000 milliosmoles, you can see that there is a very clear difference, right, between concentration, between the osmolality at one concentration and another. Due to some of the properties of conductivity, this is not always the case. And I would just draw your attention to acetic acid, where you have a weakly dissociating solution. So obviously the ability to conduct currents through something like that is a little bit tougher. So in these cases, osmolality has really clear value as being a unique, but also a really complementary measurement to something like conductivity and pH. And I also, before I move on to filtration systems, I do want to point out that we did a recent study, or actually Dr. Alois Jungbauer and his team out of Vienna did a study recently, and it was published in January, that kind of took advantage of some of those really unique properties of osmolality. This was a protein-based study, so it's not really highlighted here, but if you'd like information on the study, I'd be happy to share. Please let us know and we can provide that paper to you. But really the finding was that in trying to develop a model to predict protein concentration during purification processes, he was able to use osmolality alongside UV as a predictive measure for protein concentration. And ultimately, the idea is that if you can rely on a model using osmolality to give you insight into your protein concentration, you could potentially save a lot of time and prevent some hardships and some issues down the line in your downstream processing. So it's a really great find. We've definitely gotten a lot of interest. We just had another webinar earlier this week. So if you're interested, please let us know. We can point you in the direction of that data. So if we go back to cell and gene therapy, specifically, the last piece that I'll discuss in downstream operations would be filtration. So we know that tangential flow filtration, or TFF, is critical as far as UFDF operations within this field. Typically, due to how delicate or how fickle some of the therapies can be. So the question here is, how would osmolality testing complement these operations? How is it going to benefit? What areas or issues that are currently being experienced within filtration could be addressed by osmolality testing? And my answer is, osmolality testing is a great way to verify the completion or the quality of either your concentration steps or buffer exchange steps. So as you're exchanging buffers, depending on the number of diavolumes you're using, it's great to take the osmolality measurements at each of those stages, maybe every 2 dye volumes, to ensure that you are successfully moving from one set of buffers to another. So that's one piece, is really monitoring your concentration and buffer exchange steps to make sure that your buffers are exactly what you're expecting, to make sure that the composition of your drug substance is what you're expecting. The other piece is really interesting, and I wanted to focus a little bit more on this, is the in-process ability of osmolality testing to potentially reduce the potential for vector aggregation. So, of course, ultimately, the goal is to achieve high concentrations of viral vectors in a drug substance and ultimately in a drug product. And we know that this really lends itself to aggregation issues around filtration, especially within enveloped viruses. So I wanted to highlight, there's a study that I have cited here on this page where they actually used osmolality testing to check the levels of excipients, so check the different concentrations and osmolalities of different excipients that they knew would help to prevent aggregation within AAV2 vectors. So that's just one thought. I'm sure that there are a lot of other ways that are that can specifically aid in kind of resolving some of this pain point that is aggregation. But I wanted to highlight this one. And I also, again, ask that if you have any experience with this, or this is something that you are particularly interested in, to please let us know. We would love to get more information as we move towards some more specific investigations into this area. All right, so next we have formulation and final product testing. And really, it looks like some of you are doing osmolality testing. I'm assuming most of you are doing the compendial testing for release for osmolality testing. So what I want to focus on is actually cryopreservation and the role that osmolality testing could and should play in these processes. So cryopreservation is obviously meant to preserve the integrity of drug substances and really ultimately to preserve the critical quality attributes of the therapies that are in process. So the implications for osmolality testing of the cryopreservative solutions themselves, as well as for the formulations, are critical to maintaining the cell quality and maintaining those critical quality attributes. So here I just mentioned testing the osmolality of the cryopreserves before testing or before cryopreservation is key to make sure that you're not going to do any unintentional damage, obviously, to the therapy. But also, it's important to remember that upon thawing, this would be another great time to measure the osmolality of the formulations. So here, I just wanted to highlight, and I'm sure many of you are familiar with this, but the point of this diagram is really to just say that any time that you have cryopreservation as part of your process, it would be a really great time to consider using osmolality testing to make sure that the osmolality or any pressures that could be affecting the integrity of the cells is not changing from one step to the next. And here, I wanted to present this information because this is a question that we've gotten a few times and maybe is on your mind as well. At Advanced Instruments, we offer freezing point depression osmometers, which mean really great accuracy and precision for testing a number of different sample types. But what happens when you're testing reagents and agents that are intended to have an anti-freezing effect? So things like DMSO, or maybe not anti-freezing, but slow the freezing process. So we do have some data just to really look at the osmolality of different cryopreservative solutions, whether they're homemade, and you can see those in the blue bubble to the left, or commercially available options such as CryoStore. We really wanted to, not just for the sake of measuring the osmolality, and this is by no means to provide an idea of what you should expect for osmolality, but rather to show that even with the freezing point technology, you can still get really great performance when it comes to the precision of the measurements. So you'll see for these 5 solutions in particular, we did see pretty great precision and performance across the board. Okay, so that concludes and kind of wraps up and gives you some highlights of what we're working on and what we're hearing and what we're seeing across the field as far as the value of osmolality testing in cell and gene therapy. The goal is to dig a lot deeper. So again, we look for, collaboration and questions from you and comments from you as we move forward. But now I'd like to address a little bit of what advanced instruments can provide for those of you that are interested in introducing osmolality testing into your workflow. So here you can see our Osmotech portfolio of microosmometers. So we have the single sample microosmometer on the left. It takes 20 microliters of sample. On the right, we have the Osmotech Pro. It can run up to 20 samples in one run, and it takes 30 microliters of sample. So you can see here a kind of a list of the benefits and really the focus for these new instruments and why they're so great for biopharma. So we still have the accuracy and precision that everybody knows from advanced instruments. But the idea behind these was to provide a lot of options and a lot of flexibility when it comes to your data. So data integrity, data management, data connectivity, making sure that all of these things can be flexible and meet whatever the individual needs are of each lab and of each team. And just to give you a snapshot of all the capabilities as far as connectivity, you can see that there are really, endless ways to get data from your instrument into your network fairly seamlessly and quickly. So we found that one of these ways, if not multiple ways, are working for our customers. And we've had a lot of great success stories thus far, whether you are operating with electronic batch records and you just want that information pushed straight from the instrument into your network, or you want to view your results remotely using the web server. Again, the flexibility is here within this portfolio. And if you have questions, please let us know. And just as important as the features and the benefits of the instruments themselves, I just wanted to quickly highlight some of the services and support that we also offer along with these instruments. So installation and training, IQ/OQ validation, we have preventative maintenance, and also along with that technical service, some really great rapid technical support whenever you need it. So I just wanted to point those out. That's something that we really take pride in, not just the devices, but also the support and the service that we offer. All right, so I want to just wrap up really quickly with a kind of overview of what we've seen. And you can see that just as I broke down today's presentation into upstream, downstream, formulation, final product, this really, I think it gives a nice kind of bird's eye view of how widespread the use of osmolality testing could be within the cell and gene therapy space. So we're excited to develop some of these ideas more and to learn how you are or will be using these in the future. So again, please let us know what you think. And I would like to run one more poll. just to make sure that we have an understanding of where you are, if you need more information, if you'd like to learn a little bit more, if you want to be pointed in a certain direction, we'll have that information for you below. So please respond to that poll, and I'll give you guys a few moments. And in the meantime, I also want to highlight the fact that a lot of this really came about today's webinar because we have a brand new article that it's a review of 8 ways that osmolality testing can help improve process development and manufacturing within the cell and gene therapy space. So everything that I discussed today and then some, all 8 ways, are really broken down pretty cleanly in this article. I really would invite you to check this out. And if you have any questions, of course, let us know. So we will, I have the website listed here if you'd like to download the paper, or feel free to let us know if you're interested, and we can definitely get that out to you as soon as possible. All right, and I think I can see that our polls, great. Lots of interest in receiving additional information. So again, we will follow up, and please feel free to do the same with us. Thank you so much for your attention. I believe I will turn it over to Shweta, and we can now start a discussion and have some Q&A time.
Thank you, Christina. It was really exciting to hear all these lovely new use cases and opportunity for osmolality to be implemented across the cell gene therapy workflow. And we do have some great questions already. And I'd like to start with the very first one. How can osmolality of the transfection complex be altered to improved titer in real time, especially with PEI Pro, which has a very short incubation timing prior to addition to the production culture?
Great, thank you so much for the question. So as far as in real time, I am by no means an expert in this field. So I could definitely point you in the right direction to some of the literature that we do have in this area. But I think as far as osmolality measurements are concerned, and keeping up with real time, I'm not 100% sure if this will answer your question, but it is a pretty quick test. So it takes about 90 seconds to measure. So as we work towards getting some more studies together, this is definitely something that we are currently kind of building an experimental plan with some collaborators. And that's a great question that we could potentially ask them moving into it. But what I'd like to do is maybe even connect with you if that's possible and get some more information from you to find out the best way to address the question as we move into those projects.
Perfect, thank you. We also have another question, more for the upstream application. How can we study the effect of osmolality? Do you recommend to tune the osmo by changing only sodium, or a combination of contributing factors such as sodium, potassium, and glucose?
Sure. So, I mean, that's a great question. There's clearly a number of osmolytes and a number of solutes that are going to affect osmolality. So I would say a lot of this information kind of came from very unique publications, right? So for some of these, it was even sugars. I would say in our case, we would tend to start with the salt or start with something like sodium. So yes, to answer I think the first or second part of the question, that would be our suggestion. And kind of start with that, but also recognizing, we completely recognize that the other things, potassium and glucose, I think I mentioned that the media sugar levels, when we're looking at the sugar metabolism that affects the vector stability, is definitely affected by sugar levels as well. So if you're interested in kind of doing an exploratory study, I would say start with salt. To answer your question, to start with sodium. But it sounds like from looking at the studies that others have done, that sugars are also really key. So I think you're kind of going down the right track. I would love to hear more about all of the contributing factors that you're interested in and kind of the trends that you're already seeing. So this could be turned into something really productive as far as a DOE for looking at osmolality. So we could definitely get together and kind of discuss that if you're open to it. But thank you, that's a great question as well.
Thank you. How about something even simpler? Can you explain osmolality versus osmotic pressure?
Sure. So this is a question we get often, and I would even, maybe I shouldn't, but I would even throw in osmolarity, right? So with osmolarity, it's a measurement where you're also looking at concentration, but it's as a function of volume. And we know that temperature, pressure, those things affect volume. So with osmolality, you have that temperature independence. With osmotic pressure, excuse me, we tend to see that more when it comes to really anything involving a membrane or just filtration across a membrane of any kind, and anytime you're concerned about the osmosis through that membrane. So the osmolality, again, is going to be a measure of the concentration on either side, right? The osmotic pressure is just as it sounds, it's a measure of pressure that it would take to alter, really to alter the osmolality on either side, but to alter that final concentration for the membrane. So when we're talking about measuring, especially when it comes to bioprocessing, osmolality is a key measurement and really something that's probably more valuable as a measurement because it has that independence from outside factors like temperature and pH, or sorry, excuse me, not pH, temperature and pressure.
That's a great point because I feel more and more it's important to have that parameter that's independent of these other factors.
Right.
And this seems to be a question we always do get. Thank you, Christina.
Mhm.
Another question we have is what's the suggested target osmolality value for upstream transfection to improve the productivity?
Great question. So I guess it's not a simple answer. I would say that it depends on the type of transfection. So I'd be curious to learn a little bit more about your specific methods. I know that our first participant had asked about PEI Pro specifically. So it may be a little bit different. I don't want to give you a specific range, but for a chemical transfection, it could definitely be different from something like immunoporation or electroporation driven transfection. So I'm hesitant to give a specific amount. What we've seen is not far from a physiological range or amount of about 290 milliosmoles, maybe a little bit higher, because as you kind of saw with some of these applications, there is a trend between something, and I'll tell you it's about maybe 330, 350 milliosmoles is when we start to see some additional benefits as far as osmolality testing is concerned. So that's what I've seen, but by no means is that kind of a general amount for those different applications. So, but we do actually have some literature I can point you towards, some, depending on what your method is, if it's chemical, immunoporation, or electroporation, whatever your method, we can definitely point you to some literature that may give you some more specific insight into that range that you're looking for.
This definitely seems to be a subject of interest, Christina, because we have another question related. You mentioned chemical methods of transfection. Is there any data around other methods of transfection, such as electroporation?
Yes, there definitely is. And so we actually have them for those of you, I believe that let me check. Yes, so if you're in the offers section within this webinar, you can now download the article, and it has some really great resources there that are specific to electroporation and immunoporation. Obviously, some studies that were done that really focused on developing osmolality specifications to optimize the transfection for other types. So I would kind of suggest that once you go into that paper and you go into the section, they're broken down, each of the 8 ways. So if you go into the transfection efficiency and osmotic shock section, you'll see that there are some really great references there. And of course, feel free to ask us if you have any specifics that you need to know as well.
Thank you, Christina. Again, please feel free to type in your questions, and Christina will be more than happy to answer and help continue this discussion. While we wait for some additional questions, Christina, I have a question for you. Is there data around AAV stability and osmolality?
There is. So there's not as much. I think what we've mainly seen is really how osmolality affects the membrane or the envelope of the stickier vectors, so the retroviral vectors. I've definitely come across a lot more literature for that. What I've really seen that's interesting, and it's not exactly the same thing, but when we look at the capsids and capsid proteins and start looking at a method called the osmotic shock, that's more where we see some of the literature, obviously deriving from the osmotic part of osmotic shock, is kind of driving the osmolality of the surrounding media, surrounding solutions higher, so that you can access whatever is inside of the capsid and kind of lyse the cells, but not damage that genetic material that's inside. There is definitely literature available. We do have resources for that. Again, I would say one quick way to access those is to download the article, and then you'll see again in the same section, transfection efficiency and osmotic shock, there's some really great resources in there. And of course, we can also provide information for you that's specific to that.
Perfect, thank you. Another question that just came up. Can osmolality be used to detect low-lying infections in cultured media early on?
Great question. So I'm hesitant to just jump in and say yes, as much as I would love to. I'd be interested to know a little bit more. I think it's very likely. I think without knowing more about kind of what is causing the infection, it's hard to say, but it's definitely, we have seen instances when this is the case. And I'm also curious to know how early. I think obviously it's something where if we can help you out, if there's a more specific example of the kind of the timeline and what sort of infection you're talking about. But yes, in general, we have seen publications that have shown that the osmolality is affected to at least some extent, a noticeable amount by an infection in culture.
Perfect. And yes, absolutely. I think both of us would love to hear the answer as vehemently as yes to that question. We have another question here. As cell and gene therapy is booming and a lot of focus is being placed into this particular section of the market, how do you see changes in regulation impacting the use of osmolality per se in cell gene therapy workflow? And is there any change in focus as more therapies are coming to the market?
Yes. So I would say that because we're seeing more and more strict, it seems, regulations in this area, there is a very clear need, if nothing else, to just have more control, right? So within one process or from one process to the next, there needs to be that reliability there and accountability and consistency. So osmolality testing is great because, we've talked about the definition. We've talked about how accurate and precise this measurement can be. So as a process control, from one workflow to the next or from one run to the next, it certainly is going to be there as a really robust and stable specification. So within all of these applications that we've discussed, it's something that can be checked and monitored across the board. So we're definitely seeing tighter regulations, and that even brings when I heard the question, I even thought about as far as our products are concerned. Another thing that we've focused on, I know this year, Shweta, has been the data integrity and making sure that everyone's in compliance, and that also went into the development of our instruments in the Osmotech and the Osmotech Pro. So we are for sure keeping that in mind, not only with the ability to test your solutions and the applications, but also with the products themselves. They're definitely designed with that in mind.
Exactly. As an organization, as we continue innovating on our newer products, it's very critical that these instruments going into your workflows are able to make it easier for you to comply with these various regulations and really have a seamless transitions and seamless integration. All right. So we have another question. How are your systems developed to transfer from research process development to GMP? I think that we really addressed that question with the previous answer from Christina, that our products are really built to ensure compliance to the different regulations and different needs of customers in both, whether it be research, process development, or in GMP. We're very cognizant of these differences and want to have that product really adapt to those needs. And a big part of that really is having such webinars and conversations with customers in the industry. All right. I think we're almost approaching the end of the webinar. Christina, do you have any thoughts and comments that you'd like to share with the rest of our audience?
Yeah, sure. I really definitely appreciate the questions from the audience for sure. I think this is really critical to our team, kind of continuing to develop content. Like I said, we're in the early stages with all of this, but it certainly is exciting for us to see what's important to you guys and to see what's important within the field. So I would just say thank you so much for your attention and for joining us today. And we would love to get in touch with you and continue the conversations even after this webinar.
Thank you, Christina. And from my end, thank you everybody for joining. It's always enjoyable to have a webinar where the audience is also engaged and asking these questions. So thank you so much. We at Advanced Instruments are really looking forward to continuing this conversation on value of osmolality in cell gene therapy, as well as continuing to innovate products that can meet the various needs across the workflow, ranging from low volume to sticky samples to even the high throughput workflows within the industry. So having said that, thank you so much for joining our webinar today. This will be available on demand later. Thank you, Christina, for taking your time, and have a very wonderful day, everybody.
Bye.
Bye.